In this video, I'm going to explain a little bit more about the process of metabolism, and we'll look at how a lot of cells that are out there that need energy are going to be getting their energy from their food. And when we look at metabolism, we're looking at a set of controlled reactions that take place in cells, and they can be broken down really into two parts or two types of reactions. The catabolic reactions are where you're breaking down molecules into smaller ones, and that releases energy. So we can see polymers over here are being broken down into smaller and smaller molecules. And on the other side of this, we have our anabolic reactions, where you take these small molecules and you hook them together into larger ones. And this process is going to use energy because you're typically going to be forming bonds, and that's going to require some energy. If you think back to our covalent bonds, you're going to be basically causing those electrons to shift so that now they're circling around multiple atoms, and that's going to form that covalent bond that's very strong. And when we look at this process, we also have to consider the role of adenosine triphosphate in metabolism. And if you look at this figure here, you can see that when we pump energy into this reaction, we're going to take our adenosine diphosphate, which has two phosphates, and use that energy again to shift our electrons around, and we're going to do a phosphorylation reaction where we're actually adding a phosphate onto the end of this molecule to make ATP. This phosphate bond has a lot of energy stored within it, and you can think of it as just a battery that is out there that has a lot of stored energy available, and that energy is going to be used by the cell to actually do work. So when they're moving things around within the cell, or when they're doing anabolic reactions and creating these larger molecules, they can basically transfer the chemical energy that is stored in ATP, the cell's battery, and use that to make more of these covalent bonds and make longer and longer chains of these molecules that are out there. And what we're going to look at today is the catabolic reactions that are involved in breaking down sugar, because this is a key set of reactions when we look at metabolism in cells that are out there. And if we look at the first step of this process, it's known as glycolysis, which literally means sugar splitting. The glyco is sugar and lysis is splitting. And if we look at this first step here, glucose is a 6-carbon molecule. Remember, carbohydrates, they contain carbon and water there, and there's a lot of potential energy stored up in these bonds between the carbons there. And in a series of reactions for glycolysis, that glucose is actually split into two pyruvic acid molecules, also known as pyruvate. These reactions take place in the cytoplasm of pretty much all the cells that are out there that can do glycolysis. And when they do this process, they're going to be taking that glucose and splitting it into two, and the energy that comes out of that splitting reaction is going to be used to charge up two molecules and make two ATP molecules. During this process, the cell actually invests two ATP molecules, and they generate 4 total at the end of this series of reactions. So the cell is going to get 2 ATP molecules out of this net. And sort of the way you can think about this is, if you put $2 into a bank today and tomorrow they give you $4, are you going to keep investing in that bank? Probably. For the cells, this is an energetically favorable reaction. Yes, they do have to have some energy. They need that 2 ATP molecules, but the net, what they get out of it, is actually greater than what they put in. So it's worth it for the cells to actually do this process of glycolysis. So now we take a look at the end products. What can you do with those pyruvate molecules? Because there's still 3 carbons that are hooked together, so there's more energy trapped in those bonds between the carbons. And in what's known as the intermediate step in these catabolic reactions of glucose, the cells are going to take that pyruvate, both of the molecules that were produced from our original glucose, and they're going to transport that pyruvate into the mitochondria if they're eukaryotic cells. Notice bacteria do not have mitochondria, so they cannot do this step. Instead, they're going to be doing all of this in the cytoplasm for those cells. And in this intermediate step, the cells will take that pyruvic acid with the 3 carbons that are there, and they actually chop off or decarboxylate the molecule and remove one of the carbons from it. And when that reaction takes place, some of the energy is going to come out of there, and it's going to get transferred to what's known as an electron carrier. So during this step, no ATP molecules are actually being generated. But what does come out inside of the mitochondria matrix or the fluid is the 2 leftover carbons from the pyruvate are going to get added to what's known as coenzyme A, which is basically a carrier molecule. You can think of it as it's shuttling these carbons to the next set of reactions that are out there, where we're going to transfer the rest of the energy out of these carbons and store it up in our ATP and other energy-rich molecules in the cell. So again, within the mitochondria matrix, our leftover acetyl coenzyme A that was generated from that intermediate step is going to have those carbons get oxidized. And when you look at this next set of reactions known as the citric acid cycle or the Krebs cycle, depending on the textbook, we take those carbons that are in there and we strip the rest of the energy out of there and we oxidize the carbons. And the end product out of this set of reactions here is those carbons are going to get oxidized to form carbon dioxide or CO2 in the mitochondria. This is a waste product that our cells are producing here. And that energy that is coming out of these oxidation reactions is going to be used to recharge more of those ATP molecules. So effectively what the cell is doing is it's recharging its batteries by transferring the energy out of that glucose and into ATP. And if we look at this process, those carbons and glucose are oxidized to carbon dioxide. The first set of reactions with glycolysis, we generate 2 ATPs, and then we transfer the leftover carbons into the mitochondria where that acetyl coenzyme A is made. Again, no ATP is made there. And we drop those carbons into our citric acid cycle where the rest of those carbons are going to get oxidized to CO2. And if we look at this process, the overall amount of energy that's going to be released from our glucose is going to be used to recharge 4 of these ATP molecules. And if you look at the top of this figure here, what you see is a lot of the energy was not used in ATP synthesis. A lot of that energy that is left over from our electrons in the original glucose are transferred over to what are known as electron carriers. And again, if you look at these are vitamin-derived factors. And if you look on ingredient labels, you will see things like flavine, riboflavin, and niacin. And so those vitamins are actually used inside of cells to carry these high energy electrons that are left over from our oxidation of the glucose molecules that were out there. So there's all this energy that is being stored on these electron carriers, and the cells can get that, but they have to have a special mechanism in place in order to get access to those energy-rich electrons that are stored on these carriers. And that's what we'll take a look at in the second part of our video.